Yes, I'm an immigrant to Norway, recently, I guess, six years ago, from Canada. So I sang with the Canadians and stood up for the Norwegians last night. We've been working with some founding science from NMBU since 2015 toward using knowledge of the bacterial and microbial interactions in fish health, especially considering the Aliivibrio genus of bacteria. Aliivibrio is a very broad and diverse genus of bacteria. It's very prevalent and highly abundant in marine environments. A lot of the species have a long evolutionary relationship to marine species. One of the classical strains that has a beneficial effect that you may know of is this Hawaiian bobtail squid, who lives in symbiosis with an Aliivibrio species that causes photoluminescence and protects the squid from predation. When the moonlight is shining from above, predators can't see a dark shadow of the squid going through the water at the top. So it's a very elegant, beneficial relationship for the two species. So we have a screening program where we were using various different microbial techniques to isolate probiotic bacteria from salmonid species, especially from Atlantic salmon. And in this case, we use 2 or 3 of those Aliivibrio de novo species to microbially enhance the salmon. Those are fully characterized, and as I said, it's a very diverse genus, but Aliivibrio are the 5th most prevalent genus in the marine environment in all oceanic environments in the world. So we use the term pro-microbial enhancement because at least to differentiate a little from probiotic, which presumes in-feed treatment with live microbial material. Pro-microbial enhancement in this case is delivered via the water. In the most convenient time to the fish is when they're crowded and anesthetized before vaccination, so that's where we also implement with the pro-microbial bacteria. Fish take up bacteria readily through their skin, lateral line, and gills. Especially 2/3 of the bacteria, when you isolate different parts of the fish, are taken up through the skin. You can rapidly culture 10 to the 5 microbes from the blood as a rapid transition within 5 minutes of immersing fish in a high pulse of probiotic bacteria. Previously, these organisms and the idea about microbial enhancement in salmon was looking at classical approaches to outcompete in vitro, for instance. These Aliivibrio strains, there's beneficial Aliivibrio strains are outcompeting almost all gram-negative bacteria of fish disease, with the notable exception of Yersinia ruckeri. The publication by Øystein Klokkegg in 2021 explained the effects in post-smolt production in Norway. So our question to ourselves was, could we look at this purposeful intervention with pro-microbial strains to mitigate infection with sea lice? The reason why we hypothesized that could be so was because we saw distinct changes to the skin quality, robustness, and also the number of mucus cells and mucus production in enhanced fish. So we are, as I said, bacteriologists, microbiologists. We didn't have a lot of expertise with sea lice, so we have a collegiate relationship with Øystein Evensen's group from NMBU, who are looking at development of sea lice vaccines, and they had a ready, established and validated sea lice model. It's pretty typical. So in the first study, we took Atlantic salmon that were already in saltwater, and we allocated those to group treated some with the pro-microbial product, absolute control, and then we had a cohabitant group to look at cohabitant effect, where half of the fish were treated with the pro-microbial and half were not. They were immersed for 60 to 120 seconds. The reason why we do that is it coordinates with the time that usual to reach stage 2 anesthesia in most anesthetic baths. That's a high pulse, as I said, so they're immersed in 6 times 10 to the 8, in this case, bacteria, total bacterial count. And then we did a lice challenge 5.5 months later. So they were exposed to a single immersion with the bacterial content. The typical challenge, 30 copepodids per fish, and we counted lice 34 days afterward. The results are here in group, so you can see, but in the case of the standalone tanks with pro-microbial against the control, the relative percent protection was 63, and with the cohabitant groups it was 60. But you can see a dampening of the infection here, and also we don't know how much cross-treatment effect is going on when you cohabitate. This is just to show you the same data, but also to show the consistency. It's very well grouped. These are the pro-microbial enhanced group, and these are the controls in separate and cohabitant tanks. The second study that we did was to examine a little bit more about that cohabitant effect. So we had in freshwater, smaller fish, pit tagged. We immersed them in 20 to 30 minutes in a static bath in their own tanks. They were held in freshwater before smolting. And then 128 days this time after administration of the treatment, we counted lice 39 days and 100 days after the original challenge. So that was a generation in tank, subsequent generation of infection. Here, the control groups in tank. Unfortunately, we found this cohabitant effect to be very significant, so all groups in here ended up pro-microbial enhanced and verifiable Aliivibrio content. So we have an external control for infection control. And this is the result of the lice count on the external control at 167 days after treatment and 257 days after treatment. We were trying to look at a microbial impoverishment with halamide, but again, the common shared probiotic effect was within the cohabitant tank, 100%. This is a prolonged effect at 8.5 months. Then we looked at the stage of development there in that same trial. So we counted the number of adults and calamus and pre-adult mature female from the control versus the pro-microbial enhanced. And as you can see, there is a delayed development in the fish groups that were administered the treatment. Subsequent to that, we took lice egg strings off of the fish that were treated, raised those egg strings in hatching chambers, and looked at their propensity to reinfect when they hatched copepodids. These fish were naive, so meaning no treatment at all, and then we administered the copepodids. Then there was oh no. Okay. Yeah, so here the ability to attach of the lice egg strings raised to copepodids coming from Stembiont treated fish was much lower than the wild type. So that was also a good finding. Quantitatively and morphologically, we saw a very odd picture of the egg strings. They weren't straight. They had a sometimes curved and helical structure. This is showing at the same stage, 34 days after infection, where the egg strings were fully developed in the wild type lice on control fish. You can see the lack of pigmentation and lower development of the egg strings from Stenbion-treated fish. So the key findings here that we had a reduced lice attachment on fish that were pro-microbial enhanced just the once in the administration. That was consistent and reproducible with a RPP of 60 or more. The prolonged effect was measured up to 8.5 months, and I may have said, but that was a generational challenge, so we left the original lice challenge on fish and let them go through, we assume, 2.5 generations, and then counted again with 80% RPP in that case. So we see that this has potential for reduction in the attachment potential of offspring from Stenbion-treated fish, as well as the viability and hatching success. So all in all, it has quite a lot of good things going for it as a preventative biological treatment enhancement. In acknowledgement, you may have seen this data presented at the Lice conference in Norway in 2021 by our PhD, Marie-Stine de Blanc. We have submitted this paper to Aquaculture for publication, so you can read it in full if you like. And we had assumed that we would go on to do more epidemiological work before we would register this product for commercial use. But in consultation with the Fisheries Directorate, looking for permits to do controlled field work with the product under the research permit, we were told that the product was so negligible risk and high potential that we should just sell it to commercial breeders. So we did. We registered the product with the Environment Department, Department of Environment and Climate as a microbial hygiene product. And the next presentation with my colleague Ragnhild is to present the best possible data that we can get from use on commercial aquaculture sites. So I'm basically just here to introduce my colleague. So thank you very much. We can have questions later.